In multicellular organisms, cell division is essential for three major functions: growth, development, and repair. Through cell division, a single cell becomes two cells become four, and then four divide into eight, and so on. Cell division contains three major parts: interphase, mitosis, and cytokinesis. Most eukaryotic cells spent most of their time in the interphase section of cell division. Interphase is divided into three different sections: the G1, S, and G2 phases. In G1, also known as growth 1, the cell creates proteins, replicates cytoplasmic organelles such as vacuoles and Golgi bodies, and grows in size in order to prepare for the future phases of cell division. Following the G1 phase is the S phase, in which DNA replication occurs in the nucleus of the cell. At the beginning of the S phase, each chromosome is made of one coiled DNA double helix, also known as a chromatid. At the end of this stage, each chromosome has two identical chromatids, with replicated centrosomes as well. After the S phase comes the G2 phase, in which the cell undergoes a period of rapid growth to prepare for mitosis. The G2 phase prepares the cell for mitosis by checking the replicated chromosomes for errors and by producing enzymes that will help the cell divide. After interphase is finished, mitosis begins. Mitosis consists of four parts: prophase, metaphase, anaphase, and telophase. When prophase starts, DNA molecules are condensed and begin to form visible chromosomes in the cell. Enzymes during prophase also break down the nuclear membrane and the nucleolus of the cell. Spindle fibers also form in prophase, where they later attach to the chromosomes. In metaphase, the spindle fibers attach themselves to the centromeres of the chromosomes and align the chromosomes in the equator or the middle of the cell. In anaphase, the spindle fibers shorten and the centromere splits, which separates the two sister chromatids in the equator. In telophase, the final part of mitosis, the chromatids are pulled to the opposite poles of the cell. In addition, the nuclear envelope and the nucleolus reform, and the spindle fibers disintegrate. Finally, the original cell divides its cytoplasm and physically splits into two new daughter cells. This process is called cytokinesis.